So for the photosynthesis lab, we're going to deal with a technique called chromatography. And this one specifically will be the paper chromatography. So you're going to take a piece of chromatography paper, being very careful not to get your prints on it and to remember, which side is the top and which side is the bottom. And then with a pencil, you are going to create a line, a horizontal line, about 2 centimeters away from the base of the paper. And this line here we will call the origin. And this has a distance so far of 0 centimeters, or whatever unit you want to use. You're going to create an extract using the crushed leaves and acetone, and then you're going to use a capillary tube to place drops of that at the center of this line. So there's my capillary tube, and I'm going to use it to place four drops at the center. Okay, be sure to let this dry. Now you can take it to the next stage. We're going to take our chromatography paper and we're going to put this in a chamber. We'll call this a chamber. It could be a beaker or a bottle. And this is going to have some kind of solvent which will travel up the paper. So solvent. In our case, we are going to use acetone. So do not dip the paper completely. When you insert your paper, make sure the water level is below your origin line. It'll travel up eventually. So there again was my origin line, and here was my drops of those extract. Now the solvent via capillary action is going to travel up this piece of paper. You are going to stop this experiment once it has reached, a few centimeters from the top. So once you are there, you are going to stop. Now for some terminology. This piece of paper, we will call this the stationary phase. Stationary because it is not moving. Stationary phase. Because the paper again is not moving. The solvent which is moving, we will call this the mobile phase. The mobile phase. And this compound here, so let me draw another picture, is going to split up based on its preference for the solvent or the stationary phase. So here was my piece of paper. There was my pencil line. We still have our acetone there, but now it already finished crossing to the top. And our red dot changed a little. We might see, a green region. We might see a blue region. If it travels with the paper, so if it travels higher up, that means it's more attracted to the solvent. However, if it stays closer to the origin, that means it prefers the stationary phase. Okay, so now the line where this ends, so that would be right here in this case, we'll call this the solvent front. And this is also a distance. Let's say in this case, it's 10 centimeters. We know this is our origin line. This is the origin, which has a distance, origin by definition, 0. To calculate RF, which stands for retention factor, so we can, identify these compounds, compare them to literature values, you need to find the distance to the center of the spot of this marking, and then from the origin. So here, let's say it's 3 centimeters. To calculate RF, you would do 3 centimeters over the distance, the solvent front distance, which was 10 centimeters. That gives me a retention factor of 0.3. Now let's see for this second spot. Let's say this has 6 centimeters to the center of the spot. The RF for this point would be that distance, 6 centimeters, over again the solvent front, which is 10 centimeters. Always the same denominator. This gives me a retention factor of 0.6. Okay, so you're going to do this with your leaf extract, and here are some literature values for pigments you might be able to isolate. Okay, the next part of your lab, you're going to be inserting your extract into a spectrophotometer. And let's kind of explain how this works. Here is a cuvette or a mini test tube, and inside is going to be some kind of solution. So the solution I'm going to put inside my spectrophotometer. And what it's going to do is shoot a parallel monochromatic light. It's going to shoot some light at it, and we call this the incident light intensity. We indicate this with an I0 because it is the initial amount of light that goes through. Now the light that exits, we call this transmitted light intensity. We give the symbol here a I. Now it's possible that your compound is going to absorb some light. So I'll indicate that with this red. So it's possible that your solution can absorb light. Which is fine, no issue there. To calculate transmittance, it is simply the intensity you're left with over the intensity you started with. And then a useful equation here would be percent transmittance, which is simply I over I0 times 100%. Okay, now, and for absorbance, uses a logarithmic scale. It is going to be the negative log of T, of transmittance. So this is I over I0. So you can shoot different amounts, the different wavelengths of light through your sample and see what the transmittance and absorbance is. And this is all I wanted to mention for the spectrophotometer.